Hi there, everyone. We are so happy to have you, whether you are joining us here today or will be watching later on the Cure GABA-A YouTube channel. My name is Monica Joanna Alnacave, and I'm the founder and CEO of Cure GABA-A, a nonprofit organization with the sole focus to develop potential therapies for GABA-A variants. By partnering with world-renowned researchers in GABA-A and the Organization of Patients for Clinical Trials. Today, we welcome Alexandria Foksinska, Program Manager, and Alexandria Howard, Case Analyst at UAB Hugh Kaul Precision Medicine Institute. Alexandria Foksinska and Alexandria Howard will both discuss artificial intelligence, AI-driven drug repurposing for rare disease. Alexandria Foksinska is a Program Manager at UAB. Her background is in molecular biology and biomedical sciences from the University of Alabama at Birmingham. Alexandra's research interests include identifying drug repurposing candidates for rare genetic disorders using artificial intelligence. Alexandra also serves as the project coordinator for UAB Center for Precision Animal Modeling, as well as a committee member for the Undiagnosed Diseases Network Therapeutic Matching Committee. Alexandria Howard is the case analyst at UAB. She is currently approaching her senior year at the University of Alabama in Birmingham with a double major in genetics, genomic sciences, and neuroscience, and a minor in chemistry. Alexandria's research interests include investigating various rare genetic disorders and identifying drug repurposing candidates using artificial intelligence. I'm handing it off to you. Please help us understand how artificial intelligence can help in drug repurposing.
My name is Alexandria, and I am a case analyst with the Hugh Kaul Precision Medicine Institute at UAB, or PMI for short. And today I will be talking about artificial intelligence, or AI-driven drug repurposing for rare disease. For this presentation, I'm going to be breaking it up into 6 different topics and then finally leaving room for any questions that anyone may have. So without further ado, we can go ahead and get started with AI and biomedical knowledge. I wanted to kind of give you all some statistics. There are over 7,000 different rare diseases that impact around 300 million people worldwide, and approximately 80% of these diseases are genetic in origin, with 95% not having any FDA-approved drug treatment. And with genetic testing becoming the new norm in clinical settings, the number of rare diseases diagnosis will continue to increase. So what does that mean for us as researchers and our doctors? It means that the need for new treatment options is increasing, but identifying these treatments for these rare disorders has been known to be challenging. So to start off with, there is a limited understanding of these disease mechanisms because these diseases are so rare. And this, of course, makes it difficult to develop efficient treatment options that hit every necessary point. So also, when we look at small cohort sizes, small patient populations make it difficult to convince researchers and companies of the need for treatment, and also to gather sufficient evidence for FDA approval for these drug treatments. And then there's also inter-individual symptom variability, meaning that even though two people may have the same rare disease, their symptoms may produce differently compared to one another, which also makes it hard to develop one treatment that would fit both patients. And then finally, as we know with pharmaceutical company, there is little incentive to create these new treatments for these rare diseases because A, they can be expensive to develop, and B, they, of course, like we said before, it's a small patient population. So kind of switching gears a little bit, there's also limitations to finding new information. So as we know, there are lots and lots of scientific papers being published every year, which could rightfully so make it hard to find new relevant information that could help with new treatment development. And, since there's a lot of papers being published, a good segue into the second point is that, this knowledge tends to get forgotten or pushed to the back burner because there's so much information being published everywhere, and everything's, not being connected properly. So we can potentially be missing out on older publications that could also lend a really helpful hand to drug discovery. And then finally, another major obstacle is getting this information into a searchable format that is not only accessible, but also easy to use and navigate. So, even with these limitations, there is a way that we can reposition these FDA-approved drugs into new treatments for these diseases. And we call this drug repurposing. So, drug repurposing is like giving a second life to medications that were originally created for one condition by finding new ways they can help with other health issues. So instead of starting from scratch to develop new drugs, we can actually explore existing medications to see if they can be effective for different diseases. So this not only saves time, but it also saves money since these drugs have already been tested for safety and efficacy. So, of course, with AI, it makes this process more efficient because the AI can actually quickly go through all these scientific papers, data, and research findings to help us find these new uses for these existing drugs. So PMI has created a research consultation service that analyzes patient cases and uses our tool, Metacanarin, to aid in treatment identification and in disease characterization. So what is Metacanarin and how does it work? So Metacanarin is a reasoning engine developed under the NIH Biomedical Translator Program. And what it does is that it uses these knowledge graphs, which we'll kind of see a example in a bit, produced by the teams in the program to provide a generalized way of describing relationships between two concepts, such as drugs, symptoms, diseases. So here in this depiction, the knowledge is represented by dots, with each color representing a different category. So once you have, some structure with these concepts, you may start to see relationships forming between each concept, which creates knowledge and connections. So each piece of knowledge is unique. So for example, if we zeroed in on these two dots at the very bottom, yes. So we see that the light green concept interacts with the brown concept in some way. It also may be something more specific or even have directionality. So like in this example, we see that the light green dot inhibits the brown dot. So all of these connections that you see form this structured knowledge graph. So to kind of shake things up a bit and make it a little bit more complex, if we have this dark green dot here representing a concept, it is going to be related to this yellow dot. And then which is also going to be related to this light green dot and so on. So we can infer that through these connections that you see here, that the dark green concept is related to the light green concept in some way. And if we go even further, we can infer that the dark green concept is also related to the brown concept in some way. And these connections, like the dotted line connections, they're not considered directly related, but they are considered associated with each other based off of the other connections, which is shown here by the solid lines that are directly connected. So this makes up a knowledge graph. So I also wanted to give an example of how this may look with specific concepts. So let's say, for example, we have a disorder that is known to disrupt a cellular pathway. And then this pathway, there is a gene that is associated with it. And then there could be a drug that we have found that is known to activate this gene. So, of course, like I mentioned before, the solid lines represent a known relationship, which is, of course, backed up by evidence in the form of scientific publications. And you can also infer through this that this gene is associated with this pathway, which could also be potentially related to this disorder. Or we can also see that through its activation of the gene, this drug can impact the pathway, which also could impact this disorder. So if we, of course, wanted to, infer this information, we would have to, of course, read the publications behind the concepts in order to see how and if in what way these concepts are related to each other. So what do we need to do a metacanrin search? So we first need to identify a gene target or targets, and then we have to determine direction of regulation. So whether we want to activate or inhibit, and then whether we want to manually review any hits that metacanrin returns to us. So, as we know, AI is not perfect by any means. So there may be chances that we get false positives or something that is completely unrelated to what we are trying to look for. So that is why it is important that, my job as a case analyst is, going through all these different results to make sure that, to get rid of any of the false positives and also to really get the information that we actually do need that is helpful. So what kind of hits do we get with metacanrin? So sometimes we do get lucky, and we do get a drug that directly regulates a gene target of interest. And we call this a one hop because you hop over once and it's like a direct connection. But what happens if you have a gene or pathway that also regulates your gene target of interest? We can also find drugs that can regulate this pathway, which would then regulate the gene target of interest. And this would be considered a two-hop. So also you may be wondering, how do we prioritize one drug over another? So metacanrin uses publications to rank drugs. As you can kind of see here, you have some drugs that have 9 publications, 2, 5, 1. And the more publications that metacanrin pulls on a drug interacting with the gene target, the higher it appears on our list. So I also wanted to give kind of like a real life, case that we did have with metacanrin in action. So for this case, we had a gene, or ADNP was our gene of interest. And mutations in ADNP was believed to be caused by a deficiency or decreased amounts of ADNP protein in the body. So in this case, we want to upregulate or increase ADNP. So one of the drugs that came back was ketamine. So of course, with any results that we do get that may be of interest to a researcher or physician, it's vetted by our own clinical team that consists of, doctors, physicians, different things like that. And of course, after several discussions that we did have, we saw that ketamine was then used in a clinical trial at Mount Sinai for ADNP syndrome. So here's kind of like the papers and also the actual clinical trial where ketamine was used. So this study was the first of its class for ADNP syndrome, and it provides preliminary clinical evidence for improvements in different things like social communication, ADHD, speech, and activities of daily living. So that's really cool seeing how, metacanrin actually was useful and helpful with this clinical trial and everything. So we wanted to see, what do we want to accomplish with our findings? So we want to eventually get to the point where we can screen potential drugs in animal models. So, of course, drug screening in animal models is a vital preclinical step in drug discovery, because it also helps us evaluate the validity, safety, and efficacy of a potential drug before they are tested in humans. And then also in N-of-1 trials, which is conducted on a single patient to determine which drug works best for that patient, and also the effectiveness of these different drugs to see which one gives a optimal treatment for this patient. So, and then finally, how can you get involved as a patient community? So, it starts with you. Participation is key. So in order for us to have the most accurate and helpful information, it is important to collect data from a variety of individuals, regardless of their gene, variant, rare disease, anything like that, because we want to be as inclusive as possible when we do our research so we can benefit as many people as possible. And Also here I provided some examples that Monica has mentioned to Aleksandra and I, like the matrix surveys, the variant app, and also the citizen natural history study, which I believe are all linked on the Cure GABA-A website as well. Really want to say thank you guys again for giving us this platform to speak and give this wonderful presentation. And me and Aleksandra are happy to answer any questions that you guys may have.